chapter of Minneapolis, March program. The time's getting away from me. We're glad you can be with us. We have a wonderful program lined up for you this evening. My name is Keith Olsted. I chair the board for the Audubon chapter of Minneapolis, and I'm glad to welcome you for this evening's program. We'd like to start our program simply by asking, what have people been seeing? I've had some good luck just this last few days in finding both winter wren and brown creeper. Amazing brown creeper finding spiders under heavy bark. It's just amazing to watch them. You can use either the chat function to report what you're seeing, or you can raise your hand and we'll unmute you so that you can tell us what you're seeing. So fill us in on what you're finding out there.
Keith, you're muted.
Spacebar isn't consistently unmuting me, interestingly. So Irene Bueno Padilla reports that she saw the mountain bluebird at Como Park. That is a gorgeous bird. Lisa Thornquist found a lesser scaup. Another lesser scaup Wilmer found. Young great horned owl landed outside my window. Stayed for several minutes. That's from Dina Thorson. Eastern bluebirds fighting over nest boxes in Oklahoma, a little further south. Although we've had some overwintering here in our St. Paul golf course too, the Como Park golf course. Reports of trumpeters, common goldeneye, slate junco, dark-eyed junco, house sparrows. Isn't that amazing that you'd find house sparrows? Brad Griffin, and then northern cardinals and American robins. So you're seeing stuff that's getting ready to do some moving around. Oh, a red-shouldered hawk has returned from Marilyn and Warren Regelman. A red-breasted nuthatch, that's a good find this time of year. I wonder where you are that you found that. A couple of pine grosbeaks. I wonder if that's further north or if that's down here in the middle part of the state. Craig Thielke reports nothing exotic, chickadees and cardinals. Jim Harkness found a northern harrier. That's pretty good. Golden eyes. A red-breasted nuthatch outside his window in St. Paul. That's for Irene, outside her window in St. Paul. Oh, the Regelmans are in North Oaks. Very good find. Good find. So I think we're on the very early edge of some bird movement, and that's lots of turkey vultures in New Hampshire. Yeah, we probably won't see them here for a couple more weeks, but I've been watching to see if I can find my first golden crowned kinglet. That would be another sign of spring coming. So very good. As you're inclined, please continue to enter reports in your chat function. It's fun to see what people are finding. While we wait for more folks to come in, let me simply say that in terms of our chapter's work, upcoming meetings we'll list for you at the end of this evening's program. For now, let me say simply that our Migration Partners Committee continues to be connected to our partners down in Honduras, where they are still trying to recover from 2 hurricanes and the recent spike in COVID-19. At this point, our banding activities in Honduras are on hold. The Zamorano University folks are ready to get started in the fall, and so we're looking forward to that. but we continue to be in conversation about some other things we could get going while we wait for the banding to pick up in November or so. Our advocacy committee has been doing a lot of work. I think everyone probably has heard that we won our Supreme Court case in mid-February. We now, the case goes back to district court, and where we have applied for a temporary injunction against the comprehensive plan so that we can have a chance to do some environmental analysis of the plan's impact on our environment in the metro area. We'll keep you posted on that. We're, in the meantime, avidly pursuing fundraising to pay for our legal fees. We're also, as a committee, trying to find a way in which we can do the park board planning with a more comprehensive, holistic point of view, so that wild space and wildlife can be maintained in some of our parks, as many of our parks as possible. That's going to take some work because it's not been easy going for us. But we're dealing with the park board master plan. We're currently working on a situation at Lake Hiawatha where we're hoping for the restoration of some fairly major marshlands. We'd want you to know that we're participating in a new alliance that's emerging for the protection of wildlife in regional parks. That alliance is called the Legacy of Nature Alliance. If you want more information about that, please don't hesitate to contact me. And the Community Engagement Committee is doing some marvelous planning for major activities that will happen around World Migratory Bird Day. So watch for more information about that coming up. Continue to do some work on birdability as well for people who live with mobility or other challenges. So we have an active, busy chapter. Welcome you to support us with your contributions or to get involved and volunteer with us. Check our website for ways in which you can do that. I think with all that being said, we'll move then into our program and get right on with it. Let me introduce Katie Burns, co-chair of our Community Engagement Committee and board member, to introduce the program for this evening. Again, thanks for being with us.
Thank you, Keith. So first, Jess, I wanted to make sure that I pronounce your last name properly since I've only seen it printed. How do you pronounce it? Is it McLaughlin? Great. And what is the name of your assistant, your program assistant?
So this one is Railteen. And I actually have another one who may or may not make an appearance named Tati. So we'll see.
Great, thank you. So the program tonight, we're really excited for this program. And so I'll just give you a little bit of a summary and tell you a little bit about our presenter, Jess, as well. So what makes a species? Why are there so many more birds in some places than others? These are the questions that we can explore in many ways, but one method is comparative genomics, where the genomes of many species are compared to try and tease out patterns of their evolutionary history. From the Bering Strait to the Isthmus of Panama, join us on an evolutionary adventure to learn more about how we can study these questions and understand more about how all the stunning diversity of birds we all love has come to be. Jess McLaughlin, she, is a PhD candidate at the University of Oklahoma. Born and raised in Ohio, their lifelong love of poking things with sticks and wandering around in the woods naturally led to pursuing a career in science, starting with a Bachelor's of Science and Master's of Science at the University of Alaska Fairbanks, then to a PhD program in ecology and evolutionary biology. When Jess isn't in the lab or working on trying to get some code to run, she's enjoying hanging out with her dogs, as she is tonight, accumulating various artsy hobbies, and playing an extraordinary amount of Dungeons and Dragons. and of course, still wandering around the woods and poking things with sticks. Jess, thank you so much for being here with us tonight. I'm going to go ahead and turn things over to you, and Keith and I will shut off our cameras so you have everyone's full attention.
Awesome. All right. Oh, now you're gonna leave? All right. Oh, you need to do the screen share permissions.
Oh, one moment.
No worries.
I'm going to switch you to co-host, and I think that should address it.
Okay.
Let me know when. Let's see, it should be in effect. Go ahead.
There we go.
Excellent.
Alrighty. This up and presenting. All right. So, thank you so much for the invitation to be here tonight. I always get it. I always love getting a chance to talk about my work, particularly with, audiences outside of the usual folks at universities that I'm talking to. This is an adaptation of a talk that I've given a couple times for university audiences. And when I'm dressing it up for those groups, I've gotten to where I really like this title, Along the Speciation Continuum from North to South. But when you really get to the heart of it, there's a way simpler way to put a lot of my research interests. And it's just, why are there so many species of birds? I heard a little bit about me already, but just a little bit more. So I am basically a professional bird nerd, which is great. I am finishing up my PhD here at OU, where my main focus is on speciation genomics in tropical birds. I do a lot of work in museums. You can see me there at some of our collections at the Sam Noble Museum here. I also do field work primarily in Panama, which I'll be talking about a little bit today. You can see me with a royal flycatcher in that picture. But I was actually thinking about this earlier today. Where did I get my start, basically, in these questions? And kind of goes back to 2013 when I was a undergrad studying in Tanzania for a study abroad program, which was amazing. And I remember the first time I cracked open my field guide I'd only gotten into birding a few months before, and just looking through all of it and being like, why are there so many of these? And just having my mind absolutely blown by the diversity. And, I've gotten to travel more since then, both widely here in the US, from Alaska to Oklahoma, where I'm at now, all sorts of points in between, and also down into the tropics. And overall, like, again and again, I find myself asking the question, the main one that drives my research is, why are there so many birds? And so there's a lot of different ways we can start to get at that question. But the first thing we probably need to step back and ask is, okay, what is a species anyways? So among ornithologists, so there's a lot of different ways you can define a species, and it tends to be that people who study particular types of organisms where some definitions are easier to test than others will tend towards them. But among ornithologists, we usually stick with this biological species concept that a lot of people are more familiar with because it's sometimes in like high school curriculum and stuff. And it comes down to the basic question of, are two populations, are they reproductively isolated? Can they hybridize? And a lot of this will come down to what types of barriers prevent them from successfully creating fertile offspring that can continue to breed. And we generally break these down into ones that prevent mating, like maybe they're different colors so they don't recognize each other as the same species, or they sing different songs so they they again don't recognize each other as different species. Sometimes though, there's barriers where they successfully can mate and hybridize, but either the offspring isn't viable, or maybe the hybrid is sterile, or they're just less fit because maybe they've got an intermediate beak size and they can't really get any food very optimally. So that's so when I talk about like what a species is and how a species forms, I'm really looking at how does reproductive isolation happen? What has to happen for that to occur? And the classic way that we thought about this, and the one that it makes a great deal of intuitive sense, goes back, well into the, early 20th century. And it's this very classic idea of geographic separation, as in birds in particular, driving a lot of this process. And it makes sense. You have populations that are in different places, and either by random chance or selection for particular environmental characteristics, like in this map we see here of New Guinea, that's like this classic description by Ernst Mayr of how this process plays out. Those populations will diverge to the point where they no longer recognize each other as species and can't produce offspring. And this has been a paradigm that's been very useful. And you can see it, if you think about some of the birds you might be familiar with. There's a lot of birds where in North America, for instance, where the Rocky Mountains are an important barrier, or the Great Plains. But there's a big problem with this, as, things being geographically separate, driving reproductive isolation, and things that are reproductively isolated, not hybridizing. It's a really big catch to this. Bird hybrids are everywhere. So when I was putting together this slide, I was originally going to go and like search a bunch of stuff, and I was like, you know what would be more fun? I'm very active on Twitter, and I just asked people, hey, what hybrids have you seen? And almost all of these are ones that people submitted that they had seen in the wild, or in one case museum collections. Maybe they had banded them or whatever. And so you could see some of the normal usual suspects here in North America. You see a lot of warbler hybrids. That's not too surprising. And duck hybrids, because ducks are just always hybridizing. But we also see it in orioles. We see some more interesting combinations, like the rose-breasted grosbeak scarlet tanager that was recently confirmed as that by some DNA testing. We certainly see it, and outside of North America, we also see it in manakins and even in birds of paradise. I was really excited to have that picture sent in to use, because it just shows this is even in stuff you wouldn't necessarily expect. So what gives? The story is obviously more complicated than our brief little rundown that we've had would, lead us to believe. But when we're talking about how do you tackle such a big question, how do we even start on this? So one of the ways, and this is a way that I tend to find very useful to think about speciation, is not as discrete bits, but as a process. On one hand, you have things that they can interbreed across their whole range. They are unambiguously the same thing. A Carolina chickadee that, I have down here in Oklahoma is going to be able to interbreed with the ones at my parents' place in Southern Ohio just fine. The black-capped chickadees that I used to see in Alaska, in theory, could do just fine with the ones from your area in Minnesota. And, we're fine calling those different species, calling those the same species across the whole thing. On the other hand, we have things that are unambiguously different. Our chickadee example here, but, we can think of plenty of other examples as well. But the problem is that there's a lot of things, particularly when you start really looking and at particularly the genetics that aren't really either. And one of the cool things about this is that by looking at these things that are intermediate in the process of speciation, we can start to get a feel for what traits arrives when. For instance, do you think which comes first? Barriers like song differences or plumage differences versus, some of these more developmental differences preventing the, growth of a hybrid or whatever. What predicts, are there maybe ecological factors that predict how this process unfolds? So this gray zone in the speciation continuum, that's where I basically live and spend all my time. And by accumulating more and more genetic data sets where they're at varying points along this continuum, we can start to look for these patterns. So a quick roadmap to the talk now that we've kind of covered some of the basic. I'm going to be talking about 2 study systems that I've worked in. So the first, we're going to hang out where I spent 6 years in Beringia, going over some of my work with gene flow between Alaska and Russia and what we can learn about speciation by looking at these connections between continents. And then we're going to go south and hit the beach. Well, not hit the beach. We're going to hit the swampy lowland mangrove forests where there's quicksand, which is fun. And I'm going to talk about my PhD work, which is primarily focused on Panama. So without further ado, let's head north. So for those of you who aren't familiar, Beringia is the term we use for this region that is the Russian Far East, Alaska, and the Bering Strait. And obviously today these are separate. But as a lot of you probably know, this landmass, the continental shelf between them is actually, it's very shallow. So it's very prone to being uncovered by sea level drops in response to glaciation. And actually, the actual region of Beringia itself doesn't actually get glaciated usually, or hasn't during the Pleistocene. You have ice sheets further to the west in North America, covering most of what's now Canada. You have them extensively in Siberia, but because of various factors like ocean currents and stuff, Beringia itself has historically been a very pleasant place to be during glacial periods. And so because of this, it actually has historically served as a very important bridge between Asia and North America. Certainly humans are probably one of the big examples that come to mind, but even things like wolves, bears, moose, they all crossed this land bridge at various points during the Pleistocene. And birds are no exception. And another fact that's very cool about Beringia, and this is a little one that people are a little less familiar with, it wasn't just connected once. There were multiple rounds of Pleistocene fluctuations and sea level rises. And each time that would happen, you would get these cycles of isolation and connection, isolation and connection over and over again. And every time that happens, there's an opportunity for populations to be isolated, to start to diverge and differentiate, and then come back together. And because of that, this makes it a really interesting place to study how species form. So we know that there is in some species a degree of gene flow that's present into the present day, but it varies a lot. So for instance, we know that ducks, for instance, and this is probably not going to be a shock. Ducks, there's a lot of gene flow. This is actually a picture of one that I saw like 2 weeks before my master's defense, literally one of the hybrids that I was studying for my master's of an American and Eurasian widgeon in Alaska. And so, but it varies. Some species that, they are distributed on both sides of the strait, but don't seem to hybridize. So what's going on here? So going back to this model of looking at speciation as a continuum. So I was working with 8 different lineages of birds, and these ranged from things like blue throats and long-tailed ducks, where broadly speaking, they're the same. They're believed to be the same on either side of the strait. All the way up to things where there are different species on either side. So we got our widgeons, we got wandering and great-tailed tattlers, and we got black-billed and Eurasian magpies. And then we have stuff that's in the middle. There's differences. We can tell, oh yes, this is a Wimbrel from North America versus one from Eurasia. This is a pine grosbeak from Eurasia versus North America. This is a teal. We can tell those differences, but it's not clear exactly, like they seem to be in this middle ground. And this is based not just on, phenotypic studies, but also like some earlier studies with, 1 or 2 markers. So we can start to come up with some hypotheses. So we can first of all guess that, well, we know from previous data that these represent different ranges of how different their genomes are. And we could also guess that they probably represent wide ranges of estimates of migration. In this case is referring to, this is one of those times where being a geneticist and an ornithologist, where we use migration in both of those to mean very different things, gets confusing. In this case, migration refers to the gene flow from one population to another. And I'll try to keep saying gene flow, but it's in some of the figures and stuff, we'll say migration instead. So on the whole, we expect that as the genomes become more different, because these birds have been isolated and they have accumulated more differences, they're going to prevent hybridization. We're going to expect to see a steady drop off in gene flow. And we'd assume that this is probably going to be, again, a steady process. The thing is, that's not what we find. So looking at this, there does seem to be a relationship, but it's not the linear relationship we're looking at. Instead, we get these two discrete groups of birds that have very high rates of gene flow between their 2 populations and very little genomic differentiation. And things that are very well differentiated but have almost no gene flow. So for example, this gray bar at the bottom, that represents up to 2 birds per generation migrating from one to another, which has kind of been where we see reproductive isolation really start to kick in. We have all of these that are way in excess of it, and all of these are basically in that gray bar. And in fact, in some of them, it works out to, if you actually calculate the number of generations and like the number of birds, it's like there might be a hybridization event once every century. That's how little we're talking about here. And this is really interesting because we'd expect this to accumulate. If some of our assumptions would be that this would accumulate at a steady pace instead of this very discrete pattern that we see. And while we're not entirely sure, we think that this actually has is because of the landscape itself. So in this case, we're looking at not a smooth continuum, but one that basically has a tipping point built in. And this has a lot to do with those glacial cycles we were talking about. So every time there's a glacial period and the land bridge reemerges and those populations are connected, if a certain level of gene differentiation has not already been reached, it basically all the differences get eroded back to 0. And so over time, those that reach that level by the time the next glacial period comes around, they remain different and they continue to become more and more different. Whereas those that even just are a little bit short of that level, they're going to get reset to 0. And so over time, what you get is, instead of a smooth pattern with intermediates in the middle, you get this pattern that you see, where there's a tipping point. And this is really important to think about when we talk about landscapes like Beringia, but also other places where there's been these cycles of connection and isolation. Now we're going to shift gears. So we've looked at a system where the landscape is very important in determining how speciation is playing out and the dynamics of that process. And so Panama, though, we're getting into a little bit of different things going on here. It's a very different landscape. So first of all, before we get into exactly what's going on here, I'm going to talk about another related concept that's very key to understanding what's going on here. So just like we talked about there being a spectrum of how this continuum of speciation, there's another way to look at this. When those isolated populations come back into contact, there's also a spectrum of responses that may be reflective of where they're at along that continuum. And this is a way we can kind of observe that in action. So imagine you have two populations isolated between Eastern and Western Panama. Great. And over time, so the process here is that before Panama formed, it's a relatively young piece of land formed in the past 3 million years or so. And most of these splits are older than that. So at some point, a bird made it over what was then a strait and started a population on the other side. Great. And then when the isthmus itself actually formed, these are now connected. And so what then happens is that you have a couple different things you can predict. We can see the two outcomes we saw in Beringia, where either, they come back into contact and they don't hybridize at all, or hybridize very, very little. And that's the A that we see there, which in that case, we're showing there's a little bit of overlap between the types, but they don't in terms of where you'll find them, but they won't actually hybridize. In case of C, everything collapses like all those ducks we saw in Beringia. And then again, there's an intermediate possibility here, which is that there's some sort of hybrid zone formed, but the parental populations, East and West, still maintain their distinctive identities. So why look at this in Panama? I mean, I was already working in Beringia, so why would I be looking at this in Panama when there's potentially some of these questions could be looked at other places? So Panama is a really cool place to look at speciation. And a lot of times this focuses, on the fact that it's very biodiverse, especially for such a small country. I did the math once, and it's about a third the size of the state of Oklahoma by land area. So we're talking a very small country, and yet you have high mountains, you have lowlands, you have islands that have their own endemics going on. There's just all sorts of stuff going on. But one thing that it has that is a little less glamorous than those things is that historically, this lowland region from Bocas del Toro up in the northwest along the Caribbean and then all the way down into Darien, this lowland area was all continuous forest. And there's not any, there's rivers and stuff, but we're not talking like the Amazon or the Mississippi. These are, they could be barriers, but they're not, they're not the sort we would normally consider to be major barriers to gene flow. So you basically have this nice little natural transect that you can start to ask some of these questions about. And when folks started doing mitochondrial barcoding and some of this I've been working on this, this is an effort that started long before I was even thinking about birds. People started to notice something rather strange. So you would expect, we have highland populations that are separated. So you'd expect, if we mitochondrially barcode those and we assign them into different groups that are maybe species that we haven't recognized before, cryptic species, maybe that's, that makes sense. And, maybe some of these geographic areas cut off by mountains, maybe they'll see splits there. But when we start looking at the lowland species that occur in this broad continuous swath of forest, notice something strange. First one species, and then another, and another, over and over again. In fact, almost 20% of the birds that were sampled in multiple regions across Panama, they actually had more than one mitochondrial type. All of these are potentially cryptic species. And yet, we don't see a strong geographic barrier. So what's going on? So I dug into this, and what I found was that there's actually it's not random how these are distributed. And in fact, we can see that there are ecological traits that seem to be tied with the rise of this cryptic variation. And all of them seem to be linked to dispersal ability. For instance, hand wing index, which is a measurement that is basically the longest primary ratio to the first secondary, and it basically describes the wing shape of the bird. Lower ones mean you can't fly as well. It's much lower in the species that have this cryptic variation. If we break it down by food type, insectivores are really overrepresented. And we know that in tropical environments, insectivores don't move as much. We'll get back to why in a couple minutes. We notice when we look at where in the landscape these are occurring, what type of habitat, how open, where within the forest, these are overwhelmingly understory birds. And finally, when we look at their territoriality, they tend to be strongly territorial. So we've got some hints already that, maybe it's not the land in this case, unlike in Beringia where the landscape played such a critical role. Maybe here, with the lack of such a strong pattern, what we're seeing instead is the ecology of the birds really driving patterns of differentiation. But these are just mitochondria. And maybe mitochondria can't tell us everything about the speciation. They can be very helpful for a lot of things, but there's the whole rest of the genome too. So this is where we bring in that comparative framework again. So I looked at 10 different taxa that sampled all the way across Panama, and I think it worked out to like 280 samples or something for this chapter. And I sequenced and this is the same type of sequencing that I did for the Beringia as well, but looking at basically thousands of places in the genome instead of just one. And we knew, based on how distant their mitochondria were, that these represented a range of times. And in birds, we have a pretty good handle of what the rate of mitochondrial variation is and how that actually translates into time. And it's about 2% per million years. So for instance, down here, our plains xenops populations have probably been isolated for about 1.5 million years, all the way up to our Chiffornis up here, where they've been isolated for closer to 5 million years. And we have all these in between. And these all have different turnover places, and, but the important thing is that they seem to represent a different range of different times, probably not all arising from the same event, given that. It's not like there's a single historic factor that is likely causing all of these. And we also have a range of families, ecology, diets, all of that. So we can start to actually test some of these things. So the first thing that we're gonna so I can start to make some predictions. So first of all, I'm looking at a lot of this is looking at clines, which this is a way of actually looking at visually how a hybrid zone is actually looking at how that plays out across the landscape. And you basically go and you map the frequency of one type of one variation of a gene and the other variation of the gene, which are different in the parental populations. And then you just look at all your samples and figure out what's the proportion in each of those populations of those. And in general, the sharper the width of that is where the turnover happens, the less hybridization is going on, the more distinct these are. And so this is a really important way to test reproductive isolation. And so we would assume that in general, as time increases, and these have been isolated for longer and longer, the width of that hybrid zone, which is really what we're measuring here, is going to shrink. We're also going to expect that they're going to become more stable. So instead of like some parts of the genome moving differently across space, which is a thing that can happen, and that's really, really cool. But we'd expect that the longer you go in isolation, the less variation like that there's going to be as things stabilize and as there's more and more barriers accumulating to hybridization. And then finally, we'd expect that the genome itself, which accumulates differentiation, but not necessarily at the same rate as the mitochondria. We expect that we would expect that over time, it is going to become more and more, the higher the proportion of sites are actually going to be completely distinct between parental populations. So, did our data support these hypotheses? Nope. So, this is what the clines actually look like. I thought I took this slide out because this is for the more technical talks, but I guess you guys get to see what this actually looks like. And the first thing you can see is that these measurements are all over the place. Some of these hybrid zones literally will fit like on our campus here at OU. Like with our puff birds, like 80% of the genome is turning over within 2 kilometers. These hybrid zones are very, very narrow. Meanwhile, like our orange-billed sparrows, it's literally most of the length of Panama. And we actually had to go back and get samples from another museum collection to add on that were from Honduras and Nicaragua because we couldn't find the end of the hybrid zone in that direction. So this, and when we actually go and we map this out, we see that there's not a line here. There is not a relationship. The measurement of the fit on this is like, no, it's like it explains like 1% of the relationship or whatever. It does not help at all. This is not what's going on. We look at our proportion of genome that's fixed, same story. And we look at the variation, again, same story. It looks like time is not actually predicting any of these outcomes of secondary contact. So if it's not about time and isolation, what's it about then? So this is where I went and I ran a whole bunch of statistical models. I included time, but I also started including some other things. I included some measurements of how different their genomes were in terms of like absolute levels of genetic diversity. I included that hand wing index measurement. I included diet. And I went and I ran a whole bunch of them and fit which was best. So in some cases it was a single variable. You know, there was certainly the possibility for some of these that it would be all of them would be the best explainer. And what I found though was that generally, actually the simpler models were better. And for the variation in where the clines were happening, like how stable they were in space, the center of the hybrid zones, how stable that was predicted by the hand wing index measurements, how well the bird could physically fly, basically. Meanwhile, how much variation there was in the width. So, you know, are some parts of the genome turning over very quickly, and you're not seeing a lot of birds that have, you know, you're not seeing it spread out spatially. It just very quickly goes from one to the other. or, that measurement was how consistent that was across all the different places in the genome we looked. That was actually predicted by how well the genome was differentiated all, which hints that there's actually some genomic processes of, how genetically, how well these can potentially be interbreeding. That's what's going on there. For fixation, these were predicted by a bit more complicated models where time did play a role, which is great because it does mean we're not completely off base with our idea that time is driving the generation of variation. But first of all, it's not the sole thing, because hand wing index is, again, a very important predictor in both of these cases. And it also means that while time might help in part drive the generation of variation across the genome and across all, genetically how different these are, that doesn't mean variation in and of itself does not mean that they are reproductively isolated, which is really interesting. But what about our main thing, the actual width of the hybrid zones? What was the best predictor? And this one was really interesting because it wasn't, one of these continuous variables with numbers or whatever. It was just a very simple question. Are they getting their food primarily from insects and arthropods? Or are they getting them from plant-based resources like fruits, seeds, etc.? And in this case, it was very clear and very strongly significant that insectivores had much narrower hybrid zones than frugivores and granivores. So why does it do this, though? So there's a couple different reasons potentially at play. So like I said earlier, diet is linked to dispersal ability. We know that in the tropics, so there's always insects around and there's always fruits and seeds around. But if you're a tiny little bird and you live in the understory, insects are pretty much always going to be available to you within a very small home range. But fruits and seeds, trees aren't producing like a little bit of seeds throughout the year all the way through. Like they tend to produce them, at one particular time of year and then not be producing it the rest of the year. So a bird that is making its living solely off of fruits and seeds, it's going to have a wider home range and look for a larger area for food sources. And it's going to have to be able to fly more. Whereas if you live on arthropods, like anybody who has ever like accidentally sat on a log without looking to make sure that there wasn't anything else living on it and has accidentally sat on like a swarm of ants in the tropics, I'm not bitter about that, I swear. You will know that there are always arthropods in a lot of these environments. So you just don't have to move as much as an insectivore. So it's all about dispersal ability, right? Smaller home ranges, lower dispersal ability, get more reproductive isolation more quickly in insectivores, right? Well, not so fast. Remember, we included a very direct measurement of dispersal, this hand wing index measurement. But this isn't actually included as a variable when we're looking about predicting the width of these hybrid zones. So it might be part of what's going on, but it's certainly not the main thing. So maybe there's some other things going on. And this is where we get into stuff that I haven't been able to look at yet because it requires different types of data than what I have. But there's a few possibilities. The first and one that I'm really interested in is that maybe it has something to do with fluctuations over time. So y'all up in Minnesota, we certainly saw it in Alaska when I lived there, but you might be familiar with, there are those years when it's like all the mast years when all of the conifers in the area produce a whole bunch of cones and like all of the finches move in and just go nuts. Well, that sort of thing happens in the tropics too. You have years where there's a lot more fruit and seeds than usual. And as a result, there can be population booms in the birds that depend on them. And so there's some evidence that there's a lot bigger fluctuations among birds that depend on plant-based resources in the tropics than those that depend on insects, because the insects, they have population fluctuations for sure, but not on the same magnitude. But this is going to require more data. I have some of it. I just haven't been able to analyze it yet because I have far too much on my plate as a PhD student and a lot of research questions already. But I think this is probably a key part of what's going on, and it's a combination of dispersal and this. All right, so I'm running out of time, but we're going to wrap this up with bringing it all together. And what are the big take-home lessons from this? So, we were talking about the speciation continuum and thinking about that is this, the space in which all of these outcomes can occur. But it's important to remember from what we've seen in Beringia that landscape history itself can change the shape of this and actually change the probability of species actually being in a given part of the continuum. And this is something that's really important to keep in mind. There's going to be a lot of variations across different landscapes and that have different histories, but it's something very important to keep in mind. And then the lesson we get from Panama is that even when you don't have strong landscape factors like this, even when there's not strong geographic barriers, ecological traits themselves and the interactions between both, between birds with things like territoriality and with birds in their broader environment, with the case with diet, those can drive divergence even in the absence of geographic barriers. And so while I can't claim to have answered the question, why are birds and why are there so many birds and all of this, these two insights can bring in a different lens to start thinking about these questions in. And hopefully got you all thinking as well about some of the birds in your backyard in a different way. So just a few shout outs. I have an amazing committee here at OU. I've had great collaborators that I continue to work with at UAF. A whole bunch of museums have I've used specimens from, not just from actually not, I don't think I've used any specimens from the museum I'm actually based at, but I've used certainly a lot from the Smithsonian Tropical Research Institute, the University of Alaska Museum, Burke Museum. So thank you to all of those folks. Oh, apparently LSU is on here, and yeah. Anyways, any questions for me?
All right, for this portion, if anyone has any questions for Jess, please put them in the Q&A section and we can read them aloud to her. Looks like we're getting some thank yous, and someone commented that you have a very cute dog.
Thank you. Yeah, so I see the question, do you think hybrid birds might be more prone to disease than non-hybrids? I think that's going to depend a lot on the context. So in the case of disease in particular, it's going to depend a lot on like what the diseases are and what part of the immune, basically what portions of the immune system are going to be responsible for fighting them. Like you have different portions of the immune system that fight like parasites versus viruses and bacteria and stuff. And it's actually a really interesting question, because it actually seems like when it comes to disease, there's empirical evidence. I haven't looked into this directly, but there's actually cases where hybrids actually seem to be doing better. There's stuff going on in the Galapagos where they're looking at some of the finches there. And there's a particular like nest parasite that just absolutely like complete mortality for some of the finches nests. But it seems like hybrids that happened when there was a really bad like drought and like caused hybrid, it led to a lot of hybridization between two different species. It seems like the ones that are the intermediate actually uh do better against it, which is really wild. And it kind of just, it depends on a lot, because there's a lot of things, a lot of immune threats where actually having more variation is better. But there's definitely cases where it can be maladaptive. And it depends a lot, and it's something a lot of people are working on. I don't work on it directly as much, even though it's fascinating because it requires a lot of a knowledge of immunity that I don't have. Their question, I see a comment in the chat about if species sometimes they can't be identified, it might be a hybrid rather than a juvenile. It certainly seems that that's at least sometimes the case. I think that I think that generally you go like, while hybridization is certainly common in birds, it's not what you go to as your first explanation for sure. And so it's generally the safer bet that it's a juvenile. But yeah, there's definitely a lot of cases where the first impression of a particular hybrid bird that's been spotted, particularly if it's just been spotted in the field and it's not like it's been caught by banders, where they can really get a close look and maybe take samples for analysis. Yeah, but it's yeah, so but there's definitely probably because of that thought, it's and because it's not the first explanation, I think that there are probably hybrids that are being missed, but it's, you know, it's hard to find the balance point. In terms of albino birds, because I see your follow-up comment on that. So it doesn't necessarily seem like those are usually like one-off mutations. And in a lot of cases they're bad for the birds in multiple ways. So in a lot of cases they're going to disrupt any sort of camouflage that they have, so they might be more likely to get eaten. Particularly, you see that a lot with like songbirds in particular. And if they don't get eaten, they may not be able to find a mate because they don't have the right colorations. In terms of mutations, this is actually gets into the third reason why it's bad. So there's different types of pigment disruptions, and sometimes these are important then and can lead to the emergence of a new species, depending on what frequency it happens with. But generally, with albino birds, what's happening is that the mechanism for depositing melanin, which is the main component of most bird colors, into the feather, and it's really and so it's like just like one gene in the pathway like gets broken, basically. Like there's just one little base of DNA that gets flipped, a protein gets changed, and it doesn't work anymore. And one of the things that a lot of people don't think about that is really interesting is that actually the pigment in birds' feathers is a contributing factor in how well they can take wear and tear. So bird feathers that don't have that pigment deposited, when it's in the case of like a bird that is not normally white, so snowy owls are fine, but like in albino birds, we definitely see in cases where their feathers, they get ragged more quickly because they just don't have that you don't think, oh, the color's going to affect it, but they don't have any protection from UV rays, and so they just get really ratty, and that can also, of course, affect the bird survival. So it's really cool. I don't do much stuff on the color science angle of things, but there's a lot of really cool stuff on there. I see a question about blue wing and golden ring warbler. So that's a really interesting case. So I think a lot of it is being driven by habitat loss. But the fact that it can happen at all is driven by the fact that they are pretty young species. They split relatively recently. And they actually, it's not an issue of like genetic strength. It's just that they're genetically very similar. It's actually, there was a paper that came out on this several years ago. That's one that I seriously end up citing in like every paper I write, because they did a lot of really cool things in it that a lot of people, they were kind of like the first to do a lot of. But basically it comes down to 6 locations on the genome that are actually like strongly differentiated, most of which are linked to color variation. And like, it's like, well, this one controls pigment deposits in like the cheek region, and gives that distinctive black cheek, versus ones that control maybe where some yellow lands. And it's literally just like 6 portions of the genome. And when you have that few sites, it's really easy for when there are these fluctuations in habitat, either natural or man-made. It's really easy for those differences to be eroded. So, there's definitely, that's a complex that has a lot of studies going on in it, because it is one that is very familiar to a lot of folks, and there is some clear evidence that anthropogenic causes might be involved, but it's definitely reflective of things that we've seen in other species as well. I see the question about that I assume is both in the chat, same person in the chat and the Q&A. In terms of species moving to higher elevations, and the potential of competition for resources, are those trends create a more favorable scenario for hybrids? It depends. So you're gonna see, if it happens and like there's a closely related species that they're basically moving into the habitat of, yeah, you're gonna see more hybrids when that happens. There's definitely work being done in chickadees looking at that, between blackcap and mountain chickadees. I think there's some rosy finch work as well looking at that. But there are points though where it starts to break down, because for instance, when we're talking about higher elevations, even in like the Rockies, there's adaptation going on for higher elevation, but when you start to get into higher and higher mountain ranges like the Andes and the Himalaya, for instance, this does start to break down a bit, because when you're talking about such extreme elevations, there's the potential that hybrids are just not going to have like, they hit a point where they may not have like functional hemoglobin, basically, because there's a lot of changes that take place at these higher elevations with how they can actually carry oxygen. If you are a hybrid between a low elevation and a high elevation bird, you may end up with basically like a nonsense hemoglobin that doesn't really carry oxygen well at any elevation. So, we don't really have a lot of birds that are going to be seeing that in like in the US, just because we don't have as high as mountain ranges. a lot of our alpine birds are going to be more characterized by being able to survive like the harsh cold and wind conditions. But when you start getting into like these extreme elevations, like there are hybrids, but they may not be able to hang in there very well.
Great. If there are any final questions, we'll be wrapping up in just a moment here. So I did want to make a couple of notations for everyone. If you are interested in checking out more of the activities, getting involved with Audubon chapter of Minneapolis, as our chapter chair had mentioned in the beginning, Keith had mentioned a number of different committees that are involved in different aspects of our work, and they are meeting monthly. So you can go to the website for Audubon chapter of Minneapolis. All of the meetings are posted there with contact information on who you should reach out to if you'd like to just attend one of the Zoom meetings, just to listen in and see what we're up to and see if it's something that you might like to get more involved in. Let me take a look here. So we have our next advocacy committee meeting. That is taking place on Wednesday, the 17th at 2 p.m. And I believe the contact information for Connie or Steve is on the website, where you can reach out via email to get the Zoom information for that. Our next migration partners meeting, that should be on the website. I don't have the date right here in front of me, so I apologize, but that's a really exceptional group that is doing a lot of really interesting work. And thank you. Irene just posted in the chat box our website, so you can click right through and have that in your browser for after. Our next community engagement committee meetings, those we have she made it through all of her foraging toys and decided to participate. So that was the Goffin's cockatoo. The Community Engagement Committee, we're working, each month we're working on different planning activities and different programs, great things to do outside, all leading up to a month long of World Migratory Bird Day activities and programs. Cool stuff to do, everything from things for small children and families up to adults. So we would love to have more people involved in putting that together, and certainly joining us for some of those activities. Of course, socially distant, small groups, and bird masks, always encouraged. So as we said, those are on the website. It looks like Migration Partners is in the chat box, is on March 16th at 6 p.m. Yes, very good. So plenty of opportunities in the coming month to noodle around in the different committees of our chapter and see where you might fit in, and get a chance to meet some other bird nerds as well. All right, I think that might be it for questions, if I haven't missed any others. Jess, thank you so much for being with us tonight and tolerating the peanut gallery there at the end with the cockatoo. But this was absolutely fantastic. We really appreciate you sharing this, and we wish you all the best and the success with your upcoming defense as well.
Yeah, thank you. Yeah, thanks again for inviting me and for all the great questions. I really love every time I get to talk with, you know, kind of members of the public and field all of the bird questions and evolution questions and genetics questions. I really love all those opportunities. So
thank you. Well, have a great rest of the evening. Thank you everyone for joining us, and we hope to see you all again soon. Good night.